Hello everyone, welcome back to the Science Buffer. I'm your host, Allison Lamoureux. Today's episode will be discussing the successful launch of the ULA Starliner's first crewed mission and our feature story on a fascinating organism called the tardigrade. So kick back and relax as we dive into the newest news and interesting stories of all things science. We have liftoff. The United Launch Alliance's Starliner has officially launched and docked with the International Space Station. In the previous episode, we discussed a little bit about the ULA launch and how it had unfortunately been delayed. But now, after 2 delays, it has launched on June 5th. And it docked with the International Space Station on June 6th, just about a day after launch happened. Right now, NASA and Boeing are currently targeting for the return of Starliner on June 22nd, which will have given the crew, both the astronaut crew and the ground crew, just about 2 weeks to perform all of their tasks and run any tests that they need to. NASA will be hosting a pre-departure teleconference on June 18th, this upcoming Tuesday, for some additional details on the return mission. This is really exciting. This is the first crewed mission of Starliner and is putting them on the path toward being a partner with NASA with the Commercial Crew program alongside SpaceX. While on board station, astronauts Butch Wilmore and Sunny Williams will be completing objectives to help Starliner achieve this certification for their future flights with NASA, and they'll also be completing tasks alongside the crew of Expedition 71, who are the current crew of astronauts aboard station. During this time period, the ground crew will also be assessing performance of the Starliner vehicle and craft, and they'll also be testing and measuring 3 specific areas, the RCS thruster, the 5 helium leaks that they saw, and an oxidizer valve, all of which they found some issues with before and during launch of the craft. We're looking forward to seeing how the return mission goes and what the findings are, and hopefully this will mean another partner in the Commercial Crew program for NASA. Strange is this little creature because the whole organization of his body is extraordinary and strange, and because his external appearance, at the first sight, has the closest similarity to a little bear. Johann August Ephraim Goes, 1773, from About the Little Water Bear. This is the story of a little animal called the tardigrade that can survive some pretty extreme conditions. When I was in my senior year of college, I was trying to figure out what I wanted to do after graduation. One of the things that I'd considered was possibly going into research, and I knew if I did go into research, I wanted to do something with extremophiles and astrobiology. I found both topics to be very fascinating. For me, extremophiles are super unique and interesting because they can survive such intense conditions, especially because many of them are tiny little organisms. So it's fascinating to think about how they could possibly survive that. And astrobiology for me is an incredible way to combine 2 of my favorite topics, space and astronomy and biology, and kind of ponder the question of, is there other life out there and what could it possibly look like? So for me, learning more about tardigrades was a really awesome way to kind of combine both of those interests. Tardigrades are a type of extremophile, and they have a lot of applications in both biology, space, and astrobiology. And of course, you can't forget about how cute they are. For a tiny little organism, they are quite adorable, and as Goes pointed out in that intro quote, they very much do look like a little tiny bear. Now, for those of you who might not be familiar with what a tardigrade is, it's a tiny microorganism or animal that can survive some pretty extreme conditions, and they are a huge topic of research interest for many scientists across many different disciplines. Now, to kind of get us an idea of what it might be like to be a tardigrade, I thought it'd be interesting to try and put ourselves in a tardigrade's shoes, so to speak. Imagine you're this little organism living in the water, trapped between pieces of moss, living on a rock. It's a pretty nice life. You have some water to coat your body and food to eat that passes by pretty easily. But suddenly that water around you in that piece of moss disappears. What do? Do you find your life just slowly inching its way towards the end of it? Or do you quite literally curl up into a ball and just wait out the lack of water. That's what tardigrades do when they face some pretty extreme conditions. Now, you might be wondering, kind of from this information, okay, a tardigrade is a little animal that can survive some pretty extreme conditions, but what are they exactly, and how do they even survive that? Well, tardigrades were first described, according to Hartmut Greven, a professor at the University of Dusseldorf, by Johann August Ephraim Goes, who published the first descriptions of tardigrades in 1773. Now, tardigrades have been around for much longer than 1773. To be a little more specific, they've been around for hundreds of millions of years. But this first description can really be considered a key first step towards our research interest in tardigrades. In his description, Goes noted what a water bear looked like under his microscope, including the little claws on the ends of each of its legs. Now, to give you an idea of what a tardigrade looks like, if you've never seen a photo of it before, it's a very, very small animal, about a millimeter, maybe even smaller, depending on the species. And it has a small little head with 2 little eyes, a little mouth, and a body, which is made of 4 different segments. And each of those segments have little legs with little clawed feet at the end. And these little water bears, as they're lovingly called, can be found pretty much anywhere on Earth. They can be found in marine environments like our oceans, and plants and moss, and soil, sand of deserts, and so many other places. So they're really interesting to figure out how can they survive in such a wide variety of environments. Part of this has to do with just how many species of tardigrades there are. Scientists believe that there is upward of 1,300 to maybe even 1,400 different species of tardigrades. That's right, there's not just one tardigrade, but over 1,000 types of tardigrades. They're specifically part of a classification group called a phylum called Tardigrada. So a phylum is one of the different ways that we categorize different living beings on Earth. As biologist William Randolph Miller kind of describes, if you follow along these different classifications, you find that tardigrades are a member of the largest classification system called a domain, specifically domain Eukarya. Beneath that, a little bit more specific, they're part of the kingdom Animalia. And beneath that, they're part of the phylum Tardigrada. Now, even within their phylum, tardigrades can be even further classified as either Eutardigrada or Heterotardigrada. This is just a way that scientists categorize different species of animals into similar groups and how they evolved over time. Every species of tardigrade is a bit different, which is what makes them so interesting to study. Now, like I mentioned before, tardigrades are also extremophiles. So they can live in pretty extreme environments, both as just their regular tardigrade self, and even when they're in what's called a tun state, which we'll discuss in a little bit when we talk about how they can survive these extreme environments. To go a little bit more in depth on what extremophiles are as a whole, these are any type of organism that can live in any sort of extreme environment. These environments could include high pressure environments, very, very hot or very, very cold environments, and many other conditions. There's a whole set of classifications for extremophiles based off the different conditions they can live in. But I'll save that for another episode when we talk a little bit more about extremophiles and other research applications of them. One place that you might find extremophiles are in the hydrothermal vents in our oceans, as it's a very hot and extreme environment that not most organisms would be able to survive in. Now, extremophiles as a whole are a very unique area of research because of their ability to thrive in these conditions, and we see a lot of different fields of science studying them to figure out what applications they have and how we could use these mechanisms in other areas of research. Now, like I mentioned, tardigrades have over 1,300, almost 1,400 different species, but if you go and look into different studies on tardigrades, you might hear about a few different species in particular. I know when I was researching for this episode, there's a couple different species that popped up quite frequently that I thought it'd be important to mention. 2 of them are specifically Hypsibius exemplaris, or H. exemplaris, which was formerly classified as Hypsibius dujardini. And the other species of tardigrade is Rhamazodius varionatus, or R. varionatus. These two I found really sticking out in my research as I read through different articles and journal articles in research on tardigrades. I keep mentioning that tardigrades can survive these really extreme conditions, but how do they actually survive? I mean, they're such small organisms that you would think that something as extreme as super high heat or super high pressure would kill them very quickly. But tardigrades have a very interesting function or process that allows them to survive these extreme conditions. A tardigrade forms what's called a ton in response to these conditions and changes in their environment. A ton is basically a state a tardigrade goes into. So they typically are living their little tardigrade life, and when they find that they are encountering a change in their environment or these extreme conditions, for example, like desiccation or extreme drying, they'll turn into what's called a ton. They quite literally shrink in size and curl up into a ball. And when they're in this state, their metabolism pretty much completely stops, allowing them to survive these very harsh conditions. Now, in order to create or become a ton, a tardigrade has to sense these extreme conditions before it can enter this state. And depending on the process and extreme conditions they're encountering, will determine how long it takes for them to form a ton. This process can range in time from mere seconds to multiple minutes or more. Now, the ton formation is part of a process called cryptobiosis. There's a few different ways to describe cryptobiosis that I thought was very interesting. For example, scientists Lamoureux and Neves describe cryptobiosis as, quote, latent or hidden life, where metabolism comes to a reversible standstill, end quote, in their article, New Insights into Survival Strategies of Tardigrades. Miller, the biologist I mentioned earlier on classifications of tardigrade, describes cryptobiosis as, quote, a reversible ametabolic state, the suspension of metabolism that has inevitably been compared to death and resurrection, end quote. Basically, cryptobiosis and ton formation is a state of life of a tardigrade or other organism where metabolic processes almost completely cease, and the organism is in almost a twilight zone of life and death. They're not quite alive and being their tardigrade self, but they're also not dead. Now, this process is by no means unique to tardigrades. Other organisms do also have a similar process, as Lamoureux and Neves point out. But what's interesting about cryptobiosis is it's not just one overarching process, but rather it has different forms that take place based off the extreme condition that a tardigrade is exposed to. These different conditions will kickstart the process of ton formation, and because of that, they have their own names. There were 3 key types of cryptobiosis that really stood out to me that I thought were interesting and that lead to ton formation. The first is cryobiosis, which is caused by the freezing of the tardigrade. The second is anhydrobiosis, which is caused by desiccation or extreme drying of the water environment around a tardigrade. And this cryptobiosis form leads to a very slow ton formation. And finally, osmobiosis, which is changes in the osmotic levels in an environment. Now, this ton formation is what allows them to help to survive some of these extreme conditions, but the mechanisms are still being studied on how this process even works. There are a few different potential proteins and processes that scientists are researching currently. Some of them are including trehalose, which is a sugar that might work with other tardigrade proteins that possibly take place of water within the organism, allowing them to survive such extreme conditions, especially desiccation. Another way that tardigrades might be able to protect themselves in these extreme conditions is through a protein called damage suppressor, which binds to DNA of a tardigrade, much like body armor, to protect it from damage and destruction. Some other processes include the analysis of free radicals and how they might signal ton formation. So while researchers have a good idea of what ton formation is, the specific processes and pathways that lead to it and prevent damage to the tardigrade are still being studied, but they have some very interesting applications that we'll see in just a little bit. Now, because I keep talking about how great tardigrades are at surviving extreme conditions, it is of course important to highlight some of the ways that, or places that they've survived. Tardigrades have been sent up to space many, many times and have been able to survive some of the very extreme radiation both in space and here on Earth. In fact, they're able to survive much more radiation than us humans can, which I think is really incredible. They also, of course, have been able to survive some pretty extreme high temperatures and some very, 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 very cold temperatures nearing absolute zero. And because they've, of course, gone up to space, they've also been able to survive the vacuum of space. Because tardigrades are still being researched by many different scientists in various disciplines, there's a lot of interesting applications to what we already know about and what we're learning about them that could really lead to a lot of possible breakthroughs in different fields like medicine and astrobiology. Analysis and implementation of these different processes and proteins could be very important. For example, tardigrades' resistance to radiation damage and their ability to survive in the vacuum of space could lead to some pretty unique findings and potential future research opportunities. In medicine, their ability to survive radiation could have some pretty interesting implications for cancer research and chemotherapy. One of the key mechanisms that I mentioned that tardigrades have would be that DSUP, or damage suppressor protein. This damage suppressor protein, called DSUP, helps protect a tardigrade's DNA from damage, including from radiation, because it is able to bind and protect the DNA. In a review by Kaczynchuk, Rozymski, and Kazmarek, called The Biomedical Potential of Tardigrade Proteins, a review, they state that, quote, DSUP is a potential agent that could protect from chromosomal aberrations induced by DNA strand breaks in individuals exposed to irradiation, e.g. astronauts or cancer patients undergoing radiotherapy, end quote. There's this really interesting way that we might be able to apply this protein to human conditions. So this protein does have some very interesting potential applications. In fact, when I was initially reading through their review, before I even got to this quote, I was thinking how DSUP would be an incredible protein to study in astronauts and people who are going up into space and spending very long periods of time exposed to space radiation. And then they included this as a possible example, and I thought that was incredible. There are many other researchers who are studying these proteins and these processes to see how they might impact different fields besides just microbiology. Now, a study showed that when the DSUP protein was placed directly into human cells, DSUP genes and protein production did lead to less DNA being fractured by radiation. Now, further studies do need to be conducted, especially with the ways that the DSUP gene is taken up by cells. Kaczynchuk et al. Note that this study directly inserted the gene that produced the DSUP protein into human cells, rather than just allowing the cells to uptake it externally and then produce the protein. So it would be important to understand different ways that cells can uptake the gene. Applications to cancer research, for example, may allow for the reduction in damage to cells and DNA in cancer patients who might be receiving radiation therapies. And if the DSUP gene can even be taken up by human cells through other more natural means rather than just injecting it straight into the cell, it could be utilized for research and use for astronauts as they live and work in space to ensure that their DNA is not being damaged by this extreme exposure. Other studies on free radicals have also been noted by authors to be playing an important role for cancer research in the future. Another interesting field where tardigrades have a lot of application is in space research and astrobiology. Now, I mentioned before that tardigrades have been sent up to space multiple times in the past few decades. One of the things that they studied was how likely tardigrades are to survive exposure to radiation in space, and DSUP is likely the protein that helps protect them in these environments. Specific studies that have sent tardigrades and even their eggs in ton states have demonstrated the ability of a tardigrade to survive certain aspects of space like radiation. Now, aside from just directly sending tardigrades up into space on a rocket and exposing them to the vacuum and of space and everything that comes along with it, scientists, specifically astrobiologists, are trying to study them to gain more information on what kind of life could live outside of our own planet. Extremophiles in general are one type of organism that is a very, very large area of study within the field of astrobiology because they can live in such extreme environments. And one specific theory that scientists are looking at that might apply to extremophiles is how life came to Earth, or one potential theory for how life might have came to Earth and came to be on Earth, which is called panspermia. Panspermia is, quote, the sharing of life via meteorites from one planet to another or delivery by comet, end quote. Basically, the idea that life travels between planets and planetary bodies and possibly even solar systems. Now, because of tardigrades' ability to survive such high levels of radiation, some scientists view them as a possible candidate for the panspermia theory. Trespass and Burchell, in their Tardigrade Survival Limits in High-Speed Impacts paper, mention this and even go on to study a possible avenue of whether or not they would be able to survive this theory. In order to actually test the viability of tardigrades in the panspermia theory, Trespass and Burchell decided to test what amount of impact shock tardigrades could survive to see if they'd even be viable for this theory. Basically, they wanted to see how much impact pressure and shock a tardigrade could withstand before it was no longer a viable candidate for this theory. Their study, which had them quite literally shoot frozen tardigrades, specifically H. exemplaris, at targets in sand to see how many tardigrades survived at varying levels of speed and, because of the speed, different shock pressure levels. And they found that at higher speeds, few to none tardigrades actually survived, unfortunately. Now, they did discuss how if there are some very, very specific circumstances and locations in the solar system, tardigrades might be able to travel from one planetary body to another. But it would be important to study these actual circumstances in real life to see if these are actual viable theories. But because of that, they have found that very, very high speeds are not very viable for a tardigrade, and likely the panspermia theory would be dealing with very high speeds and high shock pressure. Now, one possible scenario they did highlight was in Europa, which is a moon of Jupiter, and Enceladus, a moon of Saturn, both of which are icy moons that are ejecting material from their subsurface oceans. And Trespass and Burchell noted that there could be potential for tardigrades to survive this kind of ejection and be able to be studied in this way. Now, both Enceladus and Europa are very key locations for the search of life on other planets, but that's a topic that I'll save for another episode. Thank you so much for joining me this week on this Science Buffer. I hope you enjoyed our exploration into the fascinating tardigrade and what research on this tiny water bear could mean for the fields of medical and space research. If you like this episode, feel free to share it on your favorite social media platform.